All right. So thank you all so much for joining. I think we'll get started now, and a few other folks might still be trickling in, but we should be able to get going. Welcome, and thank you so much for joining us today. Today we're going to be discussing and sharing our knowledge about a topic that is presumably near, but maybe not so dear to our hearts, Vulpia myuros management. So just to start, my name is Sarah Hammond. I'm the Director of Science for Ecostudies Institute based here in Olympia, Washington. For those who aren't familiar, Ecostudies is a nonprofit conservation organization dedicated to collaborative research and restoration of rare species and their habitats, with a focus primarily on prairie and oak woodlands here in the Pacific Northwest. So over the past 5 years or so, we've been watching with eyes wide open here as several non-native annual grasses have really been becoming more prominent in several of our managed and our unmanaged prairies. So this includes several bromus species and in particular 2 Vulpia species, one of which has just really outshone all the rest. Vulpia myuros has quickly become a major challenge at countless sites here in Western Washington. And from what we hear from many of you, also at several Oregon sites as well. And in some cases, this species has reached nearly 100% cover over several acres within just a few years of our initial documentation of its presence. So to address this emerging threat, we've conducted an experiment to evaluate different herbicide applications, and we've gathered information from folks across Western Washington to see what we can learn about controlling this plant. As many of you already know, it's a crafty species, seemingly able to survive and even thrive through many of our traditional treatment regimes. So today we're going to provide a bit of background on the life history of the species, share some of our findings from our experimental work, and hear from some partners about their observations and takeaways and knowledge regarding Vulpia management. After all of that, we should still have plenty of time to answer questions and ideally discuss some of the potential strategies and even partnerships around opportunities to learn more about what works to control this species. So we've recorded several of the questions, or we recorded all of the questions that many of you already submitted with your registration. And I'd request that if you do have questions during the presentation, that you submit them, write them into the chat. I'm going to be tracking that information and recording the questions. And then after we finish the presentations, we'll do our best to answer those questions. And we'll be kind of all piping in to contribute here. And I'll certainly open the floor to others to contribute your thoughts, your observations and experience. So please do stick around for the question and answer session, as I expect we'll have a lively and for sure an informative discussion. Also, I'll be sending you all a very brief survey. I promise it's the only thing we'll send out after this. But a very brief survey after the workshop, just to get some of your feedback on the topics that we presented today and any additional research or monitoring needs that could better inform our work going forward, and again, identify opportunities for collaboration. So please take a minute to respond to that survey when it arrives in your inbox. Okay, finally, please, if you haven't already, mute your microphones just during the presentation so we can limit the amount of background noise during the presentations. When we open the floor, I'll ask that you, if you didn't type in a comment or a question, if you want to raise your digital hand, I'm pretty sure that's a pun. But if you want to raise your hand, once we get into the question answer session, we'll get after that. So without further ado, I'd like to introduce our first speaker, Erin Mao. Erin is a Prairie Science AmeriCorps member with Ecostudies Institute, where she has assisted with several research and restoration projects over the past year. She'll be introducing to us some of the life history characteristics of Vulpia myuros, particularly those that make it such a good invader. After Erin, we'll hear from Melissa Habenecht, who is our conservation science project manager. Melissa has led this Vulpia control research project and will be sharing some of the results from the study. And then I'll hop back on after Melissa's presentation to introduce our guest manager presenters from our partners to hear their perspectives on Vulpia management across different sections of Western Washington. Okay, with that, Erin, you can take it away.
Okay, I think I'm sharing my screen.
Yes.
Okay, thank you. Okay, so what is Vulpia? Vulpia myuros is also known by its common name of rattail fescue. And before reclassification, Vulpia species were classified under the Festuca genus. The Vulpia were moved into their own genus because their flowers are cleistogamous, so non-opening and self-pollinating, and they have an annual growth habit versus Festuca, which are perennial and have chasmogamous flowers that do open up to facilitate cross-pollination. This is a grass that's native to much of Europe and the Mediterranean, but it is becoming a fairly aggressive invader in other parts of the world, including Australia and the United States, particularly in no-till wheat fields and the grassland ecosystems of the Pacific coast. Vulpia is a winter annual grass, meaning that it germinates in the winter and then undergoes rapid vegetative growth in the early spring, which allows it to outcompete some of the native species that emerge later in the season. Vulpia myuros is going to grow in a kind of spreading habit, and it forms these dense monocultures as it fills in the interstitial spaces between other vegetation. These areas of dense growth are going to form deep layers of thatch as the Vulpia sets seed and senesces, which then inhibits seedling establishment, germination, and growth of native plants. And the picture on the right is a really great example of that dense layer of Vulpia myuros thatch that formed at one of the sites that we have done some of our control trials at. So why is Vulpia becoming such a problem? Along with that really spready growth habit that allows it to aggressively invade in the open ground and interstitial spaces, it is adapted to a warm, a little bit drier habitat coming from a Mediterranean climate, meaning that it's likely to become more of a growing challenge in land management under climate change. Soil moisture has become a critically limiting resource for both native annuals and native perennials as they compete with invasive annual grasses. And these grasses, such as Vulpia myuros, are drought tolerant as they typically reach peak flowering several weeks before most other functional groups and will rapidly draw moisture out of the soil to complete most of their life cycle before the onset of summer drought conditions. And a recent 2021 study conducted in Oregon and Washington prairies found that Vulpia myuros occurred at much higher average cover than native annuals due to this direct competition for water. The Vulpia seeds are also fairly small and lightweight, so they attach easily to boots, animals, and machinery to be pretty easily and widely dispersed. This can intensify infestations that are already established, in addition to spreading seed to new locations. There's also evidence that contaminated seed supplies containing Vulpia have been a contributor to its spread here in the Pacific Northwest prairies and worldwide. The first documented case of Vulpia in agricultural fields in Denmark occurred in the late 90s in fields of Festuca rubra that were being grown for seed production. And additionally, 20 allelochemicals have been found in Vulpia myuros, many of which contribute to phytotoxicity of the surrounding plants. So herbicide, fire, and grazing have been used to control Vulpia with mixed results based on the timing of the treatments relative to the life stage of the plant. So going forward in this presentation, we'll talk about the results of several different herbicide treatment trials that we have done on Vulpia myuros. And with that, I will hand it over to Melissa.
Hi, I'm going to turn on my camera just for a minute here so you can put a face with my name and with this presentation. Okay, so now that we know a little bit more about Vulpia's biology and what a growing problem it is, I'm going to talk about what weapons we have to use against it, and namely chemical warfare. So we did a project in collaboration with Center for Natural Lands Management, evaluating different chemical control options for Vulpia myuros. So some background information. Over 2016 to 2018, Vulpia was accidentally seeded through contaminated native fescue seed onto 2 CNLM sites that are undergoing conversion from agricultural uses to restored prairie. And so just to give you an idea of the magnitude of this contamination problem, we've had Washington State Department of Agriculture test native fescue across several seed lots, and they found anywhere from 60 to 220 Vulpia seeds for every pound of Romer's fescue. And so if we scale that up to what that looks like in restoration, to if we're seeding 4 to 6 pounds of native fescue, that means that we could be distributing up to 1,000 Vulpia seeds per acre. And that is more than enough to wreak some havoc. And it did at these 2 CNLM sites. So the manager there, Saunders Freed, came to us and said, okay, I've got this Vulpia problem. Can you help me kick it? And we said, yeah, sure. And so we set out a project exploring a whole suite of different herbicides. So looking at both post and pre-emergence, as well as interactions among those herbicides. And so how we chose our herbicides, we selected agents that were showing promise in other studies. So chemicals that managers in the region were using that seemed successful on Vulpia. And we also scanned the literature to see what was working in other systems. And another criteria we had is that we wanted our herbicide to be compatible with grazing because it's a tool that we're using more and more at our restoration sites. So here I'll be sharing results from that research trial, but then I'll also be stepping back and looking at results from multiple studies, also looking at herbicide control across the Puget Sound. And so to guide our research, we set out with 3 primary questions. And the first one was, how effective are different herbicides at reducing Vulpia? Our second question, can we combine some pre and post emergence that might work better together than any single herbicide will work on its own? And third, we were worried about the cost of restoration and the impacts that it might have on the native species. So we also wanted to look at that. And so for our experimental design, we selected 3 pre-emergent herbicides, 3 post-emergent herbicides, and we also had untreated controls. And those were in a full factorial design. So here I've got my pre-emergence. We decided to go with Plateau, Dimension, and Esplanade. And so this is our pre-emergent control, which means that no pre-emergents were applied. And then we also have our post-emergence here. So we chose Reward. We also chose 5% vinegar. So just to give you an idea, that's the concentration that you might find on your salt and vinegar chips. And we knew that 5% vinegar was having some success at JBLM. And so we thought, hey, what would happen if we doubled it? So we also tried out 10% vinegar, and we had controls for that as well. And we ended up with 16 different treatments, including 1 treatment that had no herbicide applied whatsoever. And so what that looked like on the ground, we have our 2 experimental units here. Each of these are about 7 to 8 acres. And so we took those experimental units, divided them into 4 equal areas, and applied our post-emergence on those areas. And in one area, no post-emergents were applied to serve as control. And so within each of those 4 blocks, we further subdivided into 4 more slices, these treatment strips, and to those we randomly assigned our pre-emergent agents. So I want to give some background on the site, so not to give too much away, but site details do matter. And so Deschutes Prairie, it was used as an equestrian center for at least a decade before CNLM acquired it. And it was seeded with contaminated fescue in 2018. And here we focused on a 7-acre unit that was really heavily infested with Vulpia. As you can see here, this is just big fields of Vulpia thatch. And so in more recent history, kind of in the life of this study, in 2019, it was burned by prescribed fire and then later seeded with forbs in October. And then in 2020 is when we started our herbicide treatments. So in the spring of 2020, I've got a table here just outlining the type of herbicide, the dates that it was applied, the different chemicals we use, concentration, so you can compare those to what you might be using on your sites. Also, whether we used a liquid or granular form, and finally, any surfactants if those were used. So in the spring, you might notice there are 2 rounds of herbicide treatment here, one in April and another one in May. So we had to apply 2 rounds of treatments because the first one failed. A rainstorm came in right after we got done spraying. And so that was totally not effective. Then later on in August and September, we put down our pre-emergents. And then flash forward to 2020, again, we went in the spring, applied our post-emergent agents, only 1 round this time. And then later on in September, we put down pre-emergents, and you might notice that we switched from Esplanade to Spectacle, and that was because Esplanade wasn't available in 2021, but they used the same active ingredients. So switching over to Violet Prairie, it previously operated as a dairy farm. It's really high in nutrients. And so the plants there look like they're on steroids. They're really huge. And so Violet Prairie has a longer restoration management history than Deschutes. So it was seeded with fescue and accidentally for Vulpia in 2016, and then had forbs seeded there in 2017. And the unit that we're working on there is about 7.5 acres, and it's got less Vulpia than Deschutes. And so during the course of our study in 2020, it was mowed in May. So that was to prevent seed set. And then later on in September, it was burned. And then we put down our pre-emergent agents in October. And in 2021, we followed up with some post-emergent applications on St. Patrick's Day. And then later on in September, did a second round of pre-emergence, again, switching from Esplanade to Spectacle. And then we went in and collected a ton of data to figure out what was going on. So we monitored the vegetation before the herbicide application each year, and also 6 to 8 weeks afterwards. And so the data that we looked at were Vulpia, native and non-native cover, as well as open ground. And so to collect those data, we used the point intercept method. And so within each of our treatments, we evenly spaced 3 of these meter square quadrats. And at 16 points in each quadrant, we dropped this rainbow rod and recorded the number of times a plant hit the rod. And then we categorized those hits as either Vulpia, native, or non-native, excluding Vulpia. And so when the rod hit open ground, we also noted that. And so in addition to those vegetation data, we also collected thatch depth from the middle of each plot. And so we did that because we know that a thick thatch layer can impede herbicide from reaching plants and the soil surface, and we wanted a way to account for that in our analysis. So now I'm going to dive into results, the exciting part. And so addressing our first question about how different post-emergent herbicides affect Vulpia, I'm going to be showing two graphs here looking at how Vulpia cover has changed over our study period. And I've got two graphs. The one on the left is, the one on the left is for Deschutes Prairie, and the one on the right is for Violet Prairie. And across the x-axis, I have our different herbicide. And so the two on the left are the vinegars. The 10% is in red, the 5% is in blue, followed by the control and reward treatments. On the y-axis, I have Vulpia cover, and the units are not intuitive, so they're based on the method that we used. But what's really important here is to look at how the bars across the different treatments compare to each other. And also we want to look at whether the bars are staying close to zero. So that would suggest that Vulpia levels are staying consistent. They're not changing. They're around zero. Whether they're increasing or going up positive, or whether they're decreasing or going down. And also these bars stand for standard error. And so if you look across these two graphs, we see really different patterns. So at Deschutes, there wasn't much change in Vulpia cover. All of these are really close, pretty close to zero, except maybe in the reward treatment, Vulpia cover increased just a little bit. And so I ran some ANOVAs looking if there were statistical differences across these treatments, and no surprise, no differences. So let's, sorry about that. Okay, so now let's swap over to Violet Prairie, and we see a really different pattern. So Vulpia cover decreased across the board at Violet Prairie, including in our control treatment, which is suggesting that there's something more broadly going on that can't be attributed to any of our treatments. But if anything, maybe the 10% vinegar shows some promise, and if we had more replication, we might have seen a difference there. But as it stands, none of these treatments are different than the control. And so after seeing these data, I was really curious about what was going on with the rest of the plant community out there. So to try to answer that question, I made some plots looking at the relative abundance of different functional groups as well as open ground at our sites. And so this is 2020 at Deschutes Prairie. And here we have our different herbicide treatments, and the relative abundances of Vulpia is shown in red, followed by non-natives in orange. Then natives are in blue, and our open ground is here in gray. And so in 2020, what you might notice is there's a fair bit of open ground, very few natives here, and considerable abundances of Vulpia. So up to anywhere between 20, 40% Vulpia. And so flash forward to 2022, and wow, the natives have really increased. It looks like they were colonizing a lot of that open ground. That's great. But you do notice the Vulpia is still staying pretty consistent here. And so let's compare that to Violet Prairie. At Violet Prairie in 2020, we started off with a lot more natives. And so that makes sense because it has a longer restoration history. We've been seeding that site for longer. The natives have had a longer time to get established. And so overall, Vulpia is also lower. It just started off lower at Violet Prairie. And then we go from 2020 to 2022, and you notice that the Vulpia decreased a lot. But the problem is that it looks like that niche space was taken up by the non-natives. So non-natives increased at the same time that Vulpia decreased. Okay, so now I'm going to switch over to looking at our pre-emergent agents and what kind of effects they had on Vulpia. So I'll be showing the same graphs that I showed on the previous slide, just looking at different herbicides. So we've got our controls are in green, followed by Dimension in orange. Our Esplanade is in blue, and Plateau is this pinkish purplish color. And so looking across the two different sites, we see markedly different patterns. So at Violet Prairie, once again, Vulpia has decreased across the board at all of these treatments. And none of these are statistically different from each other. But if we compare that to Deschutes, it looks like there was a little increase in the control and the dimension treatments. But looking at the Esplanade and Plateau, those were hovering around 0, which is suggesting that the Esplanade and Plateau might be keeping the Vulpia at bay. So our final question was about different treatment combinations and if combinations of pre and post-emergent agents might work better together than alone. And so, is there a magic Vulpia killer cocktail out there that we can use? So I've got 2 plots that are similar to the others that I've shown, except they have all 16 treatments represented. And so our post-emergents are all grouped together here at the bottom. And within each of those post-emergent treatments, you will find our 4 pre-emergents. And so there's a lot going on in this graph. And so to make it a little easier, I've circled ones that I think are important. So looking at Deschutes, kind of the only 2 treatment combinations that actually decreased the Vulpia were 10% vinegar combined with Esplanade as well as Reward combined with Esplanade. All of the other ones either increased or didn't show any change. And then looking over at Violet Prairie, of course, we just see decreases in Vulpia across the board, except maybe this Plateau used alone control. And so looking at those same 2 treatment combinations, the Esplanade combined with vinegar as well as Reward, like they did decrease, but probably not more so than the other treatment combinations we showed. So takeaways, what did we learn? It's complicated. There is no one treatment fits all site solution to our Vulpia problem. But it does seem that dimension and using post-emergent agents alone that don't look particularly promising. On the other hand, Esplanade combined with 10% vinegar or Reward might be worth trying. We also learned that site-specific factors can play a large role in herbicide efficacy, so things like land use history. Both of the sites here have a history in agricultural use, and so that's likely they have altered nutrient regimes as well as like high levels of organic matter. Also, we know that thatch matters, and so I didn't report these numbers here, but I did find that thatch was a significant predictor of Vulpia reduction at Deschutes. So higher levels of thatch did work against the herbicide and make it less effective. I also learned that initial plant community composition matters. So if we have a site like Violet Prairie where we go and take out all of the Vulpia, but there are a lot of other aggressive non-natives around, then you could just be replacing the Vulpia with some other problem weed. So I think it's probably a good idea to seed natives at the same time that you're applying these herbicide treatments, and particularly perennials. And finally, we learned that the timing of your treatments and how that corresponds to weather conditions that year, as well as Vulpia phenology, can also influence success of your treatments. So now I just want to sit back and take a break here on Hamilton Mountain, which is in Beacon Rock State Park, down in the Columbia River Gorge. We're working on a project to restore mountain balds there. And there is a little bit of Vulpia, but also a ton of beautiful natives like these death camas and sea blush. Okay, part 2, the meta-analysis. So a meta-analysis is a statistical analysis that looks at multiple studies addressing the same question. And so what I did here was I combined results from different herbicide trials that were conducted across the San Juan Islands. So up here at Pacific Rim Institute and Evie's Landing, also trials from Joint Base Lewis McChord, and then several sites in the South Sound. So Deschutes, Prairie, and Violet Prairie, which I've already talked a lot about, as well as Scatter Creek and Wolf Haven. And so the herbicides that I was able to compare across those different studies were vinegar, reward, esplanade, and plateau. And so again, we're going to address those same research questions, looking at the effects of Vulpia and off-target impacts. And so before charging forward, I wanted to take a moment to give an overview of the different plots that I'm going to be sharing. Some folks might not have seen these before, and I just want to make sure that I'm taking you along with me as we move forward. And so the anatomy of these plots, they're called forest plots. And so on the left-hand side, we have the different sites that we were working on. And then each site has a box and some wings associated with it. And so and also here in the left-hand column is just a numerical representation of the box and the wings. And so the box is a measure of the effect size, and the wings are the 95% confidence interval. And so the effect size is a measure of the difference between treatment and control groups. And it's really cool because it can standardize for the different methods that studies use to measure their response variables. And so this line here at 0 means that there's no effect of your treatment. Negative values indicate, in this case, the way I have it set up, that our treatment is decreasing Vulpia or whatever other response variable you might be looking at, whereas positive values means that our treatment is increasing that response. And for each study, if the confidence interval does not overlap 0, then it suggests that it's significant. So for example, this dot here at JBLM has a really strong, significant effect of the vinegar on Vulpia, negative effect. And here at the bottom, we have an overall score that combines all the different studies and produces one effect size and confidence interval to give us an idea of overall significance. Okay, so now looking at our questions, looking at the effects of different post-emergence on Vulpia, I'll first start with the reward. And so looking across these different studies, we can see that on JBLM, this confidence interval isn't intersecting with 0. It looks like the reward did have a negative impact on Vulpia. But looking at these other studies, it seems that some lean positive, some lean negative, so really inconsistent and weak effects. None of those other confidence intervals are, they're all overlapping 0. And then looking here at the bottom at our overall score, again, not a significant result. It's suggesting that reward not particularly effective on Vulpia across all studies. And looking at the vinegars, on the other side of the screen here, we can see same thing, JBLM getting some strong, significant effects, while other studies are seeing some like weak and inconsistent effects. For both vinegars, it looks like overall not terribly effective. Now we're going to look at those pre-emergent agents. And so the effects of esplanade on Vulpia. Looking here, we can see that PRI seem to have some strong effects of esplanade on the Vulpia. It looks like VP and Evie's Landing were also leaning with negative effects. And of course, Deschutes Prairie is the oddball and actually showed maybe a little marginal increase in Vulpia. But overall, it seems that we do have a significant result here if you care about that kind of thing. So it does look like overall esplanade is quite effective on Vulpia. But compare that to plateau, all of these are intersecting with 0. Yeah, really weak, inconsistent effects there. So flash forward, let's look at impacts on natives. So looking at the reward, looks like all of these are overlapping 0, some positive, some negative. Overall, not really having strong impacts on natives. And here with the vinegars, it looks like Deschutes Prairie might be showing some positive impacts of 5% vinegar on natives, interestingly. But in general, across the board, no significant impacts of these different herbicides on natives. Okay, and now checking out our pre-emergent agents. It looks like scanning through here, it looks like Esplanade not having any significant impacts on natives, maybe even leaning positive a little bit. And Plateau, same thing. Okay, takeaways. What did we learn here? First of all, seems that consistent with our trials at Violet Prairie and Deschutes, it seems like Esplanade might be a good starting place. So it's showing efficacy across multiple studies and appears to have some minimal off-target impacts. However, I do want to point out that we don't have species-specific information for these off-target impacts. So here we lumped together annuals and perennials in the analysis. So we really need to tease those apart and look more closely at different species. And finally, I found that post-emergence weren't terribly effective on their own, but they can be used in combination with Esplanade, and that seemed to work well. So a lot of people to thank in this project. Thank you to the monitoring team for going out there day after day and collecting all of these data. So we had intern from Evergreen, as well as AmeriCorps members. I'd also like to thank CNLM, so Saunders and his crew, for all their work putting the treatments out there and helping pull together this project. I'd also like to thank JBLM and the San Juan Islands teams for contributing their data for the meta-analysis. And also big thank you to Wolf Haven and Scatter Creek for site access. And this project was funded by Western IPM, our Integrated Pest Management Center. And so I also would like to thank them for that. And that's all that I have.
All right, thank you, Melissa, and thank you, Erin. We are now going to shift quickly over to some of our partners. Please remember to post your questions in the chat as they come up. We've just had one or two come in, so don't hesitate to share any of your questions there. And so to get started with our partner contributions, I'm going to jump to Gina Smith from Joint Base Lewis McChord, who's going to share with us some of her experiences working with Vulpia there. So go ahead, Gina, take it away.
Just bring over your share. Do you want to share?
Can you hear me?
Just barely. It's pretty quiet. We can hear some background noise, I think.
I'm also hearing background noise I don't think is coming from me. So
Okay, just a reminder for folks to put on their or to mute themselves if they're not asking a question. I think that'll take care of it. Okay, go ahead, Gina.
Okay, I'm going to see if I can share my screen. Okay, people see that? Hello? Okay, thanks. Okay, so yeah, my name is Gina. I work for JBLM Fish and Wildlife. I wanted to do a very simplified version of the study that Melissa was talking about here on JBLM because it's also a plant that we are struggling with a lot. So basically going into this wanted to like I said, do a scaled down version and see if we got any interesting results and if it was something that we wanted to invest more resources in. So we looked at the same 3 pre-emergence that ESI did, and then also followed up with a reward treatment. Our study location was on Johnson Prairie, which is in the RTA, and that is just about 10 miles southeast of Olympia. So I had originally planned on having both of our study sites burned, but those of you who do prescribed fire probably know that often things don't turn out the way you want. And I ended up having half of my plots unburned, which sort of led us to have some interesting results here. So the unit down south here that we did burn, so this unit was burned very late in the season, in the end of September. So it was a very mild burn. We had good thatch removal, but hardly any impacts to moss. And then this unit up north that we did end up burning, or sorry, did not burn at all, that had burned previously in 2019. So it's not like it had 10 years of thatch build up, maybe 2 or 3 inches of thatch on the ground there. So this is just a brief overview of our results here. This is the average coverage of Vulpia in each of the plots that to start. So we have pretty high amounts of Vulpia here, mid 80s to mid 90s. So I went and I followed up after 4.5 months to look and see what these units were looking like after our herbicide application. And for Plateau and Esplanade, we got really good results, both in the burned and the unburned plots. So 100% control at 4.5 months. Dimension, as Melissa had alluded to, a little less enthusiastic about the results there, but still okay after 4.5 months. I followed up at 8 months, and we started to see a little bit of reinvasion in the burned units only, which was interesting, at 3%. So still pretty good control there. Dimension continued to have more reinvasion. And after 10 months, so this was this July, I went back and did a follow-up. We're seeing a little bit more reinvasion in the burned units with Esplanade and Reward. It seems to be a pretty good combination. It sounds like still 100% control there after 10 months. And that is in the unburned unit as well. So I know those are a little bit different results than what other people have been finding. And again, with the Plateau and Reward in the unburned units, almost 100% control there as well. A little bit of reinvasion in the burned units. And Dimension at this point is 17%, 23% reinvasion. I'm expecting that to only go higher. And I'm planning on following this up, continuing to monitor these plots to see how long this will be held off. I remember reading a study that said something like 95% of the seed bank was not viable after 1 year of Vulpia. So my thoughts here is if we could prevent invasion for 1 year, then we could, hopefully do a little bit of touch up here and there and see how things go moving forward. And then I have some photos here of just what one plot looks like. This is in our 100% control. This is one of the burned units, so using Esplanade. And as we can see here, to start, this is before treatment. We have a lot of thatch here. This is all Vulpia. 4.5 months after, we've got a lot of reinvasion, this lime green color around the edges here, but no reinvasion. 8 months, we have some perennials starting to show up. We've got some Lomatium and some Cairexes, but no Vulpia yet. And then finally, this July, this is what this plot looked like almost a year after this initial photo here. So no Vulpia yet on the interior, a lot of bare ground, which I'm a little concerned about because, as Melissa alluded to, got to see what's going to fill in that gap. It's probably not going to be native species. But you can see all this Vulpia up here, very happy. And then a quick look at the unburned plot. So again, a lot of thatch to start off with at 4.5 months. Not really seeing any green coming in. You can start to see in the untreated area some Vulpia starting to emerge there. At 8 months, we've got some fescue popping up, but no new Vulpia filling in the gaps. And again, at 10 months, some happy looking fescue, but still no Vulpia. So I was happy with our results here, and I'm planning on scaling this study up another site on JBLM, doing some more replicates, hopefully to get some statistically significant findings. Definitely want to monitor the effects on native species. This photo here is a picture of a plot that was treated with plateau that had some Collinsia bloom in it, which I was really surprised to find. That's a native annual that we have here. Don't know if that was a happy accident or what, but I want to look a little further into that. We also have camas popping up, all sorts of stuff. But I didn't take any preliminary data on what natives were here before, so I don't know what's missing from this plot. So definitely want to focus on that. And then I also want to look at some other exotic annual grasses we're having issues with on base, including anthoxanthum and a lot of the bromus species. So that is currently in the works. That's all I have.
Thank you, Gina. All right, we're gonna quickly move on to Adam Martin, who's gonna speak about some of his observations, I think primarily in the San Juan Islands, is that right? And maybe on JBLM as well?
Yeah, both. Can everyone hear me okay? Cool, and then people see a PowerPoint slide. Okay. So first, I'll try to be real quick with this. So this first slide, last September, we scaled up and did a few acres of esplanade treatment on a burn, and this is in TA15, which is sort of in the eastern part of JBLM. It's one of our big focused areas. The photo on the right is what it looked like this spring. I didn't have a pre-treatment photo, but before that, all the interstitial space was Vulpia. So just right away, that our esplanade treatment worked really well. What we had for plots, we only had 4 plots. We had 2 in a control and 2 where we treated, but the plots are 625 meters squares. So we don't have lots of sample, but they're big. And so the arrows are just showing the direction change, and this is percent cover based on point intercept. And so the big thing is this is just overall annual cover, which includes Vulpia, but there's other annual grasses we are interested in getting rid of, and invasive annual forbs. So overall annual cover. So it was really effective treatment, which was great. It was done in between 2 big rain events. So there's a ½ inch of rain, and then the treatment happened in the 2-day span before another ½ inch of rain came. This was at the end of September. And we're also interested in, there's some idea that it might be negatively impacting camas and fescue cover. And thankfully, the control plots weren't burned. So you'll see with the fescue, you see these big decreases in fescue, but I think it has more to do with when you have almost 60% cover in a plot and it burns, fescue takes up to 5 years to get back up. So it looks shocking, but I'm not too worried about that. And the camas cover increase was exciting because there was some worry that the esplanade was inhibiting camas. So overall, really promising. I think we'll continue to use this going forward on JBLM. And now let's go a few 100 miles north. So on the islands, so what's sort of a sad bonus in the South Sound is we don't have a lot of native annuals to worry about. But in the North Sound, on the islands in particular, is one of the last holdout of native annual forbs, and they're also being impacted by. We have 8 or 9 invasive annual grasses up in the islands. So we have all the bromes, we have both Vulpias, and then we have hordeums. So it's a sort of a different beast. So on Hope Island, well, this is Jones Island, but so we're doing work on a few small islands up there. And on Hope Island, we're trying to figure out how can we manage controlling the annual grasses and not impacting the native annual forbs too much. And so we looked at reward and esplanade, glyphosate, and then fusillade and esplanade. And these plots, they're box plots. So basically, you just want to see how much overlap or not there is between the plots. And so overall, the fusillade and esplanade didn't work very well. And so we had high native cover because it didn't really kill anything. It didn't kill the annual grasses or native cover. And then the glyphosate and then the reward and esplanade were about as effective for both. There was more species coming up in the glyphosate than in the reward and esplanade. So either could work. Glyphosate has its own issues. There seems to be a group of weeds that really enjoy glyphosate areas. And so you start to get new problems. So I think we'll probably keep going with the reward and the esplanade. And so how we're scaling that up is, so how we're trying to manage this issue of the native annual forbs is we go out and we try to map out where all the nice patches of prairie are and then where all the really invasive patches are. So the green areas are really, really high quality bulbs. The fuchsia areas are predominantly native annuals, and then the red areas are all mostly annual invasive grasses, except for there's one patch in the forest that's something else. But for the most part, this is how we're doing it. So now we're going to scale up, and then we have focused restoration treatments on each of the areas that don't have native annuals but have high invasive annual grasses, and they're treatable. So this is what we're doing on Hope. And this is how we're going to try to do some of the other small islands up in the San Juans on Jones Island, and maybe up on Marin State Park and other areas where we're trying to get a hold on this. So really exciting. I think the challenge is going to be how we can use pre-emergence and not just inhibit our native annuals. So I think that's going to be the big challenge and then the time span that it takes, everything's slowing down by 10 months to almost a year with feeding and things like that. So it'll be a fun adventure. That's what I got.
Thanks, Adam. Appreciate that perspective of what's happening up north as well. All right, and to wrap up our partner discussion or partner observations, we're going to invite Saunders Freed from Center for Natural Lands Management to share some of his experiences working with Vulpia at a bunch of different sites. So Saunders, if you're here, you can take the mic.
I'm here.
Great.
Yeah. Sorry, I'm dirty from the fire. I had to do mop up. Anyway, yeah, Vulpia has been a problem for us. I don't have any cool slides, so I'm just going to talk for a second, and then we'll get on to something better. So from what we've seen, we're having less of a problem on our more native, higher quality sites with Vulpia than our ag conversions. Our ag conversions with higher fertility have been completely overwhelmed by Vulpia. So all these ag conversions, so the Deschutes and the Violet sites, we these were under NRCS contract, and NRCS contract, their rules are first you glyphosate. That worked out great. Then you put down your first seeding of fescue was our plan. Well, that fescue had those Vulpia seeds in there. And so those Vulpia seeds filled the gaps between all the fescue that established. A year later, after we've treated with herbicides over that establishing stand of fescue with both grass specific and forb specifics, because the grass specifics like clethodim and fuselier don't kill fescue, so you can treat over the fescue while it establishes. Well, it also doesn't kill Vulpia, is what we found. And so we're treating over it, and basically Vulpia is just thanking us for creating more space and going to seed, and everybody's happy and everyone's fertile because it's covered in manure. And then our job was then to seed forbs into that unit. And that's the next step in the NRCS contract. And I'm like, well, we just wasted, 80 seeds per square foot with no establishment of native anything, because the Vulpia and the fescue blocked everything out. And I mean, it was even, some of this was, our fires weren't great when we did our treatments. Also, I got to say that I think our fires were in October, and they were wet, and so it didn't burn off all the thatch, and our esplanade treatments didn't hit the ground, which is why we went. And then some of our sites, I don't even think we burned it. We did granular because of that fact. We didn't have, a clean slate, and so we went granular, hoping it'd just find its way to the earth. Anyway, it was fraught with problems. And then we our first step was always, let's do, when do you spray grass? Well, around here we spray grass in the spring. Well, I think we need to spray this grass in the winter, because that's when it's emerging, and not wait till spring when it's already been growing for 4 months. And that's, we might, if we redid this study with a good burn, and because we know anecdotally on base, after a good burn, you throw down esplanade and you get zero Vulpia coming back up. You also get zero any other native annual forb coming up. So that's your trade-off with esplanade. So today we burnt Deschutes in 2 of those blocks. One of them I'm going to esplanade, and the other block I'm going to hit with clethodim this winter to try to catch it at an early growth stage. And the fescue in there is, and it's now 5-year-old fescue. It's established really well. I don't think I'm going to kill it off with a 0.5% clethodim treatment, but I think I can kill off a seedling Vulpia with a 0.5% clethodim treatment. Last winter, Pat and I had this same problem, because all the fescue we're putting out now, even today's crops that they're taking off of BFI, have Vulpia in them. Okay, so there's still seeds in every single crop coming to us, and we're putting out on the landscape, which is tough. Anyway, so we are gonna we did a January, I think late January or maybe early February, when we were seeing a little, the germinate Vulpia having a bright green carpet in some of our, and these are conversions as well with higher fertility. And I'm like, I know what's going to happen if we don't do anything. So let's do something. And we had, our 2 days of sun in that month in January of this year. And we sprayed it with clethodim 0.5%, right? Which supposedly fescue survives, our native fescue. And so those had massive reductions in the amount of Vulpia that was there the year before versus this year. And the year before, I'm sure it did great, because we're missing these treatment windows, and so it's going to seed and producing more. And so it's, theoretically it should have been worse than last year, but this year it was 90% less than the year before. And that was a January treatment. So this year I'm hoping we can start implementing some before the end of the year treatment. So like once it starts greening up and germinating after fall rains start, maybe we get a couple days in November of sun, and we can hit it with a 0.5% and punch it in the face, and then again follow that up, because it seems like 2 treatments is thorough, because the grass is so damn small too. It hides in this matrix of other plants. And so it's really, it's not like it's just standing out there saying, kill me. It's hiding under all the other bushes and sticks and stuff. And so we're finding more treatments is better. And the label says you can hit it twice in a season with clethodim at 0.5%. So we're going to try and do 2 treatments before like even spring happens, when we start spraying for our typical grasses. That's all I have to say about that. I don't know if it'll work. I wish we were doing some monitoring, because money didn't allow that. And so now this is going to be anecdotal, what happens when we do this at Deschutes. But I'm hoping maybe this year we could find some money and then monitor for when I do it next year at a whole bunch of other sites that are invaded with Vulpia, and get something that's actually would be reliably passable to other people. But we also know that from Elaine's work down in Oregon, that if you're not concerned about your fescue, if you're not concerned about your grasses, 0.5% clethodim will kill Vulpia when applied at the right windows for, because she was able to knock it out of some sites. But no, wait, she used 1%. That was it. So she bumped her percent, killed off her fescue, but definitely took care of the Vulpia. And so had good success with that. We don't want to do that. We want to keep our fescue, especially in these nice stands. But that's always an option. Moving forward, I've found on these conversions, do not do an NRCS contract, because that year of letting your fescue establish is not necessary. You should glyphosate the crap out of whatever you're doing if it's a conversion, and then seed everything at once, because the natives are competing with Vulpia when seeded at the same time. You can't put them in after Vulpia has had a year, right? Because now you're giving them that Vulpia year head start, and then it's going to germinate before your native seed. Even if you put your native seed down, it's going to germinate first and then fill the gaps, and then you're screwed. So we've had super great success when we're just glyphosating, and then bang, you throw down your whole seed bank of natives, and then you're still going to have Vulpia, because we have Vulpia in our fescue, because we want our fescue too. But it's not overwhelming our natives when you give them the same chance to make it. So we're seeing really, like, Cadmus is a good example where we're just glyphosating for years. And Tumwater West, we did the same thing. We glyphosated for a year, and then dumped all the seed at once. And I see the Vulpia plants that came up that year, but we also have a seed bank now of all the native annuals that also survived and went to seed this year. And then our perennials are starting to come in, and our perennials usually make it, so, and are competitive with the Vulpia, if seeded at the same time. So that's another piece of the puzzle. Anyway, that's all I got.
Thank you, Saunders. All right, there's my mute button, wasn't working. Thank you, Saunders, for your input there. That's really helpful. And it brings up that additional treatment option of clethodim that we weren't able to test. So, and Adam just put in the chat as well that's another treatment that they've had success with for annual grasses. Yeah, so thanks for bringing that to the table. I want to, go ahead.
Just one more thing, seeing Jake sit here.
Yep.
Yeah. Okay, you go. Take it.
Yeah. So a couple folks, I want to get to some of the questions that came in with registrations, and there were a couple questions about grazing, and I would just want to highlight that we have been tracking Vulpia cover in an area that's been grazed by Jake Yancey, who's on the call. And what we've seen so far is, well, at this one site, it's not set up as like within a strict experimental design. We're just tracking change over time here. At this site, we sprayed the Vulpia patches with 10% vinegar, and the site was grazed. And Jake, I might ask you for specifics on the timing, because I know I didn't check that out, but early spring grazing, I believe it was April. And we found in those plots, I'll remind you that the 10% vinegar alone for some of the other sites we've worked at was not necessarily terribly effective. But here, we don't know if we can attribute it to the vinegar or the grazing, but I'm assuming it's largely due to the grazing. We saw that the Vulpia decreased by 5 times in those plots. So there's 5 times less Vulpia a year after grazing than there was before. Sorry, a year after the grazing plus the vinegar. So that's something to consider if you're trying to avoid certain chemicals and you have options to graze. Jake, is there anything you want to add about the grazing or any of your observations there?
I think the main difference with the grazing compared to spraying is because in this test plot, we were able to use a herd of approximately 40 head of cattle that we were moving in about 2-acre paddocks. And we really used it as a prescription. And so we did not move them until they had targeted the Vulpia and kind of removed that layer. And we grazed fairly harshly. It wouldn't be what we would normally do on a standard paddock, but we were really doing it with the target of removing those fine grasses. So until that target species was removed was when the cattle got rotated. And so we truly used it. We didn't move the cattle until that target species was removed, or what I best knew the target species was removed.
Thank you. Yeah, it's a very specific prescription, targeting that species. So it's encouraging that, we actually showed quite a bit of promise, and something that I think is worth investigating, at a larger scale at multiple sites. So that is another kind of future question around Vulpia management and the utility of grazing as one of the treatments, especially in areas where folks are not able to use or don't want to use herbicide. And there were quite a few questions about off-target impacts. I want to highlight, and especially related to seeding, I want to highlight that the pre-emergent herbicides really are aimed at killing any seeds that are in the seed bank. And so generally, if you have native annual seeds and native perennial seeds in the seed bank along with the Vulpia, you're likely going to kill many, if not most of them as well. As Adam and Gina have seen, some of the areas that were treated at JBLM actually have had some native annuals coming in, which is surprising and encouraging. But I think that's one area also that we need to consider, both what species are seeded in and when, so that you're allowing enough time for the pre-emergent herbicide to do its work on the Vulpia seeds that are there and any of the other annual species that are there, non-native annual species. The documentation for Esplanade in particular states that it can be anywhere from a couple months up to a year of kind of persistence in terms of impact in the soil. A lot of the work, though, testing that herbicide has been in dryland ecosystems, so it may have longer persistent, and usually those are have lower organic material in the soils. That allows for that longer term persistence. As far as we know, there haven't been studies in west side, west of the Cascade systems, where it's wetter, higher nutrient soils in many cases, higher organic material relative to kind of high desert systems. where those chemicals are likely to be broken down much more quickly. And so we might not have that year-long impact from the pre-emergence. And so there may, but we don't know exactly what that timing is, or, what that might look like. But presumably, there may be possibility of seeding after a few months after the treatment, and still having success with annual seeding. So that's something that may need to be tested. Adam, you had a comment or question?
Comment. So we were doing some restoration work on Fort Casey, which is on Whidbey Island, and there was an accidental application, pretty sure, of Esplanade, and it controlled everything for like almost 2 years.
Oh, wow.
And we saw a similar thing on PRI, where Plateau was effective for almost a year, and then Esplanade, it was like almost 2 years. So it was like, but then it was great. Like year 3, things started popping up. It was confusing to understand what happens there. And then with the annuals, it seemed like the big seeded native annuals did pretty well. Like that's what was coming up. It was Lupinus bicolor, Collinsia, Plectritis, and then a lot of the small seeded annuals definitely didn't come up. So we're curious. I think we're going to try growing out some of the native annuals in treated Esplanade to see if it's consistent, or it was just lucky, or we missed it, or something like that. But just some more context on the native annuals, and then on time frames, at least up in it's also a lot drier in the North Mountain than it is down here. So
Thanks, Adam. Saunders, you have a comment?
Yeah, we did another field at Violet with Esplanade last year. And actually it was we Esplanaded it 2 falls ago, and it was on an NRCS contract. And so we were forced to seed it also the following year. And so it was about 12 months, and we drill seeded it, hoping to break the pre-emergent crust, they called it. When I talked to some of the other people about, is it going to kill our seeds? And we put down, not as much annual as we usually do for like a butterfly site. But we put down a lot of annual. And so in comparison, and you can't compare site to site just because soils are different and such, but none of the Collinsia or Plectritis came up, period. So I'm wondering if the effects of Esplanade lasted longer, and then towards the end of summer, all the late season annuals did pop up. So Colomia, Gilia, bang, they went crazy. But still, no Collinsia and no Plectritis, which is troubling for a butterfly restoration site. Also, some of the perennials were showing up, so that's good. But we also had to, like I said, we drill seeded that, and that's not my preferential restoration machine because I hate that sunbun. Sucks. Anyway, so we tried, but we had to otherwise, because the fescue is all fat and big, and so we had to try and cut that seed into the ground because there was no burn there, right? Which again, I think is, we have methods that work best, and then we're always forced to work these side channels around them, and then things just don't work well. And so, trying to seed without a burn is really challenging, and then add on an Esplanade treatment the year before, and and now I'm just like, have my hands up in the air going, I don't know what the heck just happened, but we can look at it, because of course nobody monitored it first, so now we don't know what really is we can remark on. I can tell you what I saw, but like I can say also that when I did the seedings at every other site that I didn't Esplanade the year before, Plectritis and Collinsia do freaking fabulously and it was this year. So like there's something happening at that site specifically, whether it be the drill seeder or the Esplanade, that totally prevented those couple spring annuals from coming up into that unit.
Yeah, so that's something to consider with choosing the different species that are going to be seeded in. At least it sounds so far, there's some good anecdotal evidence that those early season spring species at some sites do not establish well after even a year after Esplanade, but some of the later season species might. So again, I think another a valuable question is the species specific responses to these pre-emergence, to see if we can figure out what the limiting factors are and how long after these treatments it's safe to seed these species in. And there's some evidence in dryland ecosystems of experimenting with some seed coating methods. I'm not sure if that would be effective over here on the west side, but that's another area we're really interested in pursuing to see if we can protect some of those native annuals with some different types of compounds to coat them, like some kind of charcoal cover, basically a coating that can help absorb some of those compounds and make it so that seed can sit over a year and be protected and be able to germinate without being affected by the herbicide. So there may be options to get some of those native annuals established within that 1 to 2 year period while the Esplanade or whatever the other, most likely Esplanade is doing its thing. So things to consider. We don't have solid answers yet for West Side systems, but I think that's another potential promising avenue. I want to quickly see, we have about 10 minutes left, a little bit less, to see if there are any questions or comments from our Oregon partners. We relied heavily on Washington data for this presentation, but I know a lot of folks have been working with Vulpia down in Oregon as well and might have some really great observations or suggestions or thoughts. So if there's anything that folks want to contribute, that would be great. And while people are considering if they want to speak up, I'll also mention one, or just bring up quickly, there are some other questions about fire. And I know we talked a decent amount about fire within the different treatments that have been applied. But, I think fire alone might be really tricky as a Vulpia control treatment, just because it seems that the seeds seem to do just fine. If you're burning in the fall, if the seeds have already dropped, they're going to be able to germinate after the fire. So if you're using fire, or if you want to use fire, it has to be very carefully timed to be able to burn when it's dry enough to burn and carry the fire, but before those Vulpia plants actually go to seed. But also most likely it's going to have to be used in combination with an herbicide treatment. So you can help to clear out some of the thatch and have kind of open space, especially for the pre-emergence. Yeah, and Mary brings up, there's a few comments here. Mary Linders, thank you for identifying the need to look at moss cover as well. That's definitely an issue on a lot of the prairies, at least up here in Washington. And so, thinking about some of the challenges or limitations for these treatments is going to be important. And Mary, did you want to go ahead with a comment or question?
Yeah, if I could share my screen for a second. We did some mapping of Vulpia on a bunch of sites last year, and it's really pretty interesting to look at. And I know one of the things that I'm really curious about is, or really interested in having folks consider is not going, you can see across these different sites. So the dark brown areas are where our 25 by 25 meter cells where we found Vulpia to be common. So that was kind of like basically solid cover over at least a 10 square meter area. And you can see that there's areas that have it and areas that don't. And I would certainly, and this is 6 different sites. And so I would really encourage folks where you've got areas that there's little or no invasion of Vulpia, consider staying out of some of those areas in terms of restoration until we know how to manage for this species, because you can really see how strong some of the patterns are on these sites and the pattern of invasion. Thanks.
Thanks, Mary. Yeah, that's pretty compelling and really helpful information to have, seeing how it can move across the site. And yeah, some treatments might make it worse if we're not careful.
Yeah, well, I know there's been a lot of folks talking about talking about the fact that there's that the mow lines for fire are actually really spreading a lot of Vulpia. So, and I don't know, what other observations people might be seeing in terms of where it's showing up kind of in force. So consider.
Saunders, you had your hands up?
Yeah, I'd just say that it's interesting to see the maps from the RHA that were recent, and everything we burned in 2017 exploded with Vulpia, because that was the first crop where it popped over, and we were dropping fescue with that in it. But I also think, again, we don't have answers yet, but we know some things hurt that plant, and so we should be, if you're going to burn something, you should be following up with treatments anyway, whether it be seeding, some spraying, blah, 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 down the line. So, I think we're going to go to like SOP of booming out our burn breaks for Vulpia and non-native grasses, just to keep them, because we are, they love our disturbance regime. And so when we're making a burn break, we're disturbing the soil and making a place for it to find, set seed. So maybe we need to start talking about, adding some of these SOPs to our restoration actions, which it'd be simple to run an ATV with clethodim at 0.5% down your burn break, right? Bang, it's done. You just prevented a whole bunch of Vulpia from showing up. So, yeah.
Thanks, Saunders. Quickly, there's one question I want to address and see if anyone has an answer. If anyone's worked with Vulpia in montane coastal ranges. I don't think we have here at Ecostudies, but there might be folks in Oregon that have worked in that area.
Yeah, I've been tracking, and that was my question, but I've been tracking the spread of it on Mary's Peak, which is the tallest peak in the Oregon Coast Range. And it's been spreading there since about 2018, where they've been doing meadow restoration, doing tree removal to restore meadow. But it's now spreading both in pre-existing meadow and the cut unit. And it's, any case, probably a bigger question to ask right now, but but if anyone wants to follow up with me, I'd be curious. They were really limited up there because of the environmental agreement to what herbicides can be used. So it might be a little while before any pre-emergence can be used there. And there's only like 4 herbicides that are approved at this point. So any case, I'm happy to hear from if anyone has experience dealing with it in this kind of moist environment and without being able to use pre-emergence, which maybe they'll get it approved later, but it wouldn't wouldn't go into effect for a bit. Yeah.
Yeah. Thanks, Zach. That's something, if there is interest after this, to have kind of a working group or some kind of collective that can share information more quickly about Vulpia and its management, I think that might be something we can consider, because I know the issues are not restricted to these low elevation systems here in the Puget Sound. It definitely extends beyond that. Okay, we have 1 more minute. Tim, did you have your hand up?
Yeah, I was just going to add that one tactic we've been playing around with is mapping the Vulpia infestations before we go and burn the units. And so we have a solid idea of where those infestations are, and then we burn the unit, and then we're going to follow up with treatments, pre-emergent treatments, and kind of stick close to those polygons that we've mapped as a way of kind of trying to track, hit the Vulpia in the season in which it's germinating, but we have a really strong idea of where it is. So I was just going to add that point.
Yeah, that's helpful, thinking about tracking not just the cover, but the kind of where it is on the landscape, so we can identify where it's spreading and be kind of on top of that. It's an ongoing challenge, but I think you're right, really important to track as much as possible. All right, any other closing comments or thoughts? Okay, thank you so much, everyone, for your input and ideas and suggestions. We're going to be summarizing our project results in a report and a manuscript, and we'll continue to reach out as we identify new ways to collaborate to help resolve this new challenge. So thank you all so much. Cheers.